All right, thank you for tuning in to this virtual presentation. I regret that I was unable to attend Glasgow due to a couple of important personal events, a close friend's wedding and residency graduation that happened the same weekend. So I'm going to talk about the HCP-Style approach to brain imaging and imaging data phenotypes. So first covering what is HCP-Style neuroimaging data, how best to pre-process HCP-Style data to maintain its quality, and then what I mean by imaging data phenotypes. So first, HCP-Style neuroimaging data. So I mean high quality multimodal MRI data, including structural, functional, and often diffusion MRI acquired on as many subjects as is feasible. High spatial, temporal, and angular resolutions, enabling accurate neuroanatomical mapping, selective fMRI denoising, and maximum achievable accuracy of structural connectivity. And to achieve these goals really requires a 3 Tesla or higher field scanner, a 32 channel or higher head coil with optimized pulse sequences such as a VNav encoded structural images and multiband EPI. So one of the really key points of HCP-Style MRI acquisition is choosing spatial resolution based on neuroanatomical considerations. So we have here a group average cortical thickness map and the histogram of the cortical thickness map. And we can see the mean human cortical thickness is around 2.6 millimeters. The minimum thickness is 1.6 millimeters. And so, those fMRI studies with resolution that is coarser than 2.6 millimeters, will turn them low resolution fMRI. High resolution fMRI is those that have better than 2.6 millimeter resolution. Most brain imaging studies are still low resolution fMRI, but that's changing. And ideally, we want to have a resolution, better than mean cortical thickness so that we can accurately represent what's going on. So in the Human Connectome Project and Adult Aging Brain Connectome Project, or AABC, we had 2 millimeter isotropic resolution for fMRI. For arterial spin labeling, or ASL data, we were at 2.5 millimeter isotropic resolution. For our structural scans, or T1s and T2s, we ideally want to be at 0.8 millimeter isotropic or less, basically half that minimum cortical thickness of 1.6 millimeters. And we were at 0.8 for the Human Connectome Project Aging and AABC projects, and a little higher for the young adult. And then tractography is a really resolution-starved problem. So diffusion MRI resolution should be maximized to whatever you can do based on the amount of time available in the study and SNR. 1.5 millimeter isotropic resolution for the aging project in AABC, and a little higher for the young adult. So what happens if the resolution is too low? Well, this is a sort of a measure of confusion of where signal is coming from across cortical folds and different fMRI resolutions. And we can see there's quite a bit of this confusion at 4 or 3 millimeter resolutions, but at 2.5, it's decreased quite a bit, and at 2, it's pretty much non-existent. So if our resolution is too low, it can be hard to tell if an activation is on one side or the other of a fold, and the two sides of a fold may be very different in terms of sensory and motor cortex. Other considerations, it really helps to have a VNav, a navigator inside of the 3D T1 weighted and T2 weighted scans, because that can be used to improve image quality by realigning and reacquiring TRs if there's head motion. This reduces the need to rescan the patient or even throw out whole subjects. For fMRI resolution, we really want temporal resolution. We want to have the TR less than 1 second. We were at 0.8 for the Human Connectome Project Aging and AABC projects, a little higher for the young adult. And this enables selective denoising. We got to be using a multi-band factor of 8 to achieve this for 2 millimeter data. And this selective denoising allows us to remove the artifacts without modifying the signal. We want to get as many minutes of fMRI as possible and diffusion directions as is feasible. Using phase reversed images for diffusion data and spin echo phase reversed images that are matched to the fMRI and ASL data is very important. And this could be used for B0 distortion correction and even pseudo B1 mapping. Using multiband EPI for the ASL data enables high isotropic resolution with multiple labeling delays. We had a few lessons learned when we have done all this with the Human Connectome Project, things we wish we had done differently. One of those is it'd be better to have had pre-scan normalize on for everything. Multi-echo NP-RAGE turned out to be more trouble than it was worth. And at least in our hands, multi-echo fMRI wasn't clearly beneficial either, although I know that's a controversial position. So we've got this high quality data. How do we maintain its quality throughout pre-processing? We really want to minimize blurring induced by preprocessing, for example, from multiple trilinear interpolations or overt smoothing. Remove the spatial effects of head motion within and between modalities. Using BBR for precise cross-modal within subject volume registration really helps with that. We want to remove all the image distortion so that each MRI image accurately represents the physical space of the subject, and then we want to precisely align brains across subjects. And to do that, the HCP proposed a new standard coordinate system that we term SIFTY gray ordinates. And so the gray ordinate standard space is a combination of surface vertices, 30,000 in the left hemisphere, 30,000 in the right hemisphere, and 30,000 subcortical voxels. And this allows us to use surface meshes for aligning the sheet-like cortical structures and nonlinear volume registration for aligning non-cortical structures. And in terms of this gray ordinate space, we have around 90,000 gray ordinates with 2 millimeter average vertex spacing and 2 millimeter subcortical voxels. So how do we accurately align cortical areas across subjects? Well, we can take area MT as an example. And it turns out a volume-based registration does pretty bad for area MT. We've got 2 different hotspots of relatively low probability, or maximum overlap isn't even 50%. It's kind of a blurry mess here. A folding-based registration on the surface does quite a bit better. We got a single hotspot of high probability, but our overlap still isn't all that much above 50%. And we've got a number of individual area MTs and individual subjects that are not lined up. But if we do what's called an areal feature-based registration, we can get up to 100% overlap, getting all of the area MTs lined up. And by areal features, I mean things like architecture, function, connectivity, topography, or the areas themselves. We talked about imaging with a fast TR, and that really helps to remove artifacts and noise from the fMRI data. So we can take a functional connectivity map here. So here's the seed location with this white sphere. Here's a cartoon time series, and this basic functional connectivity map is the correlation value of this time series for all of the other time series in the brain here. And this is without any data cleanup. And doing these type of correlational analyses are really vulnerable to any kinds of non-random artifacts in the data. They produce biases. This doesn't really look like much in terms of a neurobiological effect, but if we use spatial ICA to remove the effects of head motion, all of a sudden now we can start to see the default mode network popping out here. If we use temporal ICA to remove the effects of subject respiration, now the default mode network is made more specific. We can see there are semantic correlations. And then if we remove random measurement noise, for example, with Wishart filtering, this boosts the correlation values quite a bit and makes a nice default mode network from this single surface vertex seed here. So most connectivity studies try to remove artifacts, but it's really important to remove noise without changing the neural fluctuations of interest. And activation studies benefit as well. So you can remove biases and improve statistics by doing these types of corrections. And again, hitting these targets of temporal and spatial resolution really require multi-band EPI. So we've got our data preprocessed. What about these imaging data phenotypes? Well, what do I mean by that? This is something similar to the imaging derived phenotypes, which is something that somebody else came up with. And I happen to forget what the D stood for. So I've made a IDPs that's a slightly different acronym, and I'm going to have a slightly different take on it than they did in the biobank paper. But the basic, my take on this is using the brain's structural and functional neuroanatomy to partition measures of interest in a neurobiologically meaningful way. So that means using things like brain areas or functional networks or white matter tracts, and averaging within these neuroanatomical units to reduce the effects of random noise, improving one's statistical sensitivity, and also collapsing the voxel, vertex-wise, or gradient-wise statistics in these neuroanatomical units to reduce the number of statistical tests, which improves one's statistical power. So as an example of cortical areas, we have the Human Connectome Project's multimodal cortical parcellation, where we've got auditory areas in red, sensory motor in green, visual in blue, and default mode or task negative in the darker colors, or anti-default mode and task positive in the brighter colors, with mixed coloration, for example, here in LIPV, when we have mixing of both sensory motor and visual information. So how does this work? Well, we've got our areal borders, and we've got a myelin map where our high myelins are in the reds, yellows, and oranges, and low myelins, the blacks, purples, and blues. And this is around 50,000 data points that we see here. But we can average myelin content within each cortical area and produce a map that looks like this. And this is now only 360 data points, as there are 180 cortical areas per hemisphere. And if we just flip between these two, we can see that the pattern is really quite similar. We haven't lost very much information despite decreasing the size of our data, the number of statistical tests, enormously by 2 orders of magnitude. You can do a parcellated analysis with functional connectivity. Here's a full correlation analysis on the left inferior parietal cortex showing the default mode network. Partial correlation is a sparser analysis, but it still shows the default mode network very nicely. You can do this with task fMRI. Here we have working memory contrast. This is a language contrast. And if we compare the parcellated analysis and just a regular gradient-wise or vertex-wise analysis that we have here, they're scaled with the same Z statistic, you can see that the results of the parcellated analyses are clearly more significant than those of the dense analysis. And in fact, if you just compare, averaging, the Z-stats within the areas versus averaging the time series and then fitting the GLM, if those things were equivalent, we'd have all the blue dots along this red line here. But it turns out that if you make those average time series before fitting the GLM, you get quite a bit more significant statistics, as these dots are steeper. Functional networks are another approach that one can use, and you can consider either hard definitions versus spatial weighted overlapping ones. One hard definition of functional networks was proposed by G et al. Here. Each area or each brain vertex, voxel, or gray ordinate typically has been assigned only one and only one functional network. But that's not the only way to do it. You can use a weighted approach to define functional networks. In this case, we've found the main default mode component across 5 different paradigms, including 7T and 3T resting state data, traditional tasks at 3T, a movie task at 7T, and retinotopic data at 7T. Another approach that makes weighted functionally overlapping networks, temporal ICA is done at the group. Probabilistic functional modes, or PFMs, are done at both the group and individual levels at the same time. And also the probabilistic functional modes don't enforce orthogonality like the temporal ICA. Things must be temporally orthogonal. And these type of approaches, in this case, temporal ICA, can do a great job of pulling out interesting things. So here's a weighted functional network pulled out by temporal ICA, and here's a traditional task analysis, basically showing the same thing in this 3T task data. And each cortical area most likely participates in multiple spatially overlapping functional networks generating different specific behaviors. So this idea of a one functional network per area per point or per pointed space is probably an oversimplification. All right, so just to conclude, what kind of data is going to be produced for the Adult Aging Brain Connectome Project? What are the IDPs that we're making? Well, we've got some at the cortical areal level, network level, and the tract level. And we can consider things like the size or the surface area of these, or the volume, these different structures. We can measure architectural information like myelin or cortical thickness. Functional connectivity could be done either area to area or network to network. Structural connectivity can be area to area or based on the tracts. We can do either task activation in a traditional task, or we can just look at the amplitude of the bold fluctuations, either at the areal level or network level. Blood flow can be measured in areas, networks, or tracts, and we can look at white matter hyperdensities as well. So to summarize, we want to get high quality MRI data that enables accurate mapping to the brain's structural and functional neuroanatomy. We want to preprocess this data to preserve its high data quality and enable localization of these measures to this neuroanatomy. We want to use the brain's neuroanatomy to subdivide the data to improve both statistical sensitivity and power. And this HCP-style approach is really helpful in addressing the increased focus on rigor and reproducibility of neuroimaging results that's been seen in the field. Well, there are many people in the Human Connectome Project, in the Young Adult Project, the Aging Project, and the AABC to acknowledge who I've listed here, and I may have missed others. And I thank you for your attention.